Hi everybody, my name is Shiza and today we'll be reading this research paper which is called Dense Cellular Segmentation using 2D to 3D Neural Network Ensembles for Electron Microscopy. And the reason why I'm reading this research paper today is because I've been really interested in semantic segmentation for the last couple of weeks, and I recently made a project that segments out brain tumors from MRI scans and that used 2D segmentation. And honestly, I found it pretty valuable. So I was like, you know what? Let's take this a level up and now do 3D segmentation, which in this case is not completely 3D segmentation model, but there's parts of different models that it uses to outperform the baseline models, such as the 2D UNet for segmentation, the 3D UNet, the Deep Lab Volume 3, and other ones. So let's get into the paper. So right now, we're looking at biological electron microscopy. So electron microscopy, what it does is it basically uses an electron optical instrument that shoots out beams of electrons and that basically enlarges images of nanoscale objects, which could be in a biological sense would be cells or organelles. And the thing is over here, we have biological samples of unprecedented volume. So researchers are facing the problem of making use of this data because they want to understand what's actually happening in the image. They want to gain some insight into the image by understanding what organelles are there, what cells are there, and how their volume correlates in EM images. In this case, they're using two human platelet tissue samples. So they want to look into the electron microscopy in that sense. And they want to generate rich 3D model segmentations of tissues and their constituent cells and organelles from these electron microscopy images. And so because of this, they want to use ensembles of 2D to 3D neural networks to compute this dense cellular segmentation. So as we mentioned, biological researchers are using this electron microscopy to image the structure of cells, organelles, and their constituents at a nanoscale. And we're getting an advancement, a rapid advancement in technologies that are using electron microscopy to visualize these sort of images at it. And these images have a gigavoxel image volume, which means that they're very high resolution. But at the same time, there's also many challenges because we're right now, if we're using manual segmentation, we're basically training humans to identify salient image features. And it's really hard to find that structural detail, especially at a nanoscale, across microscopic tissue regions. And it's overall, as it says here, infeasibly expensive and time consuming because it's just going to keep piling up and up if we have more demand for the segmentation, and humans can't keep up with this because of how time consuming it is, how error prone it is, and how challenging it is. Well, in that case, it's obvious that we need to leverage new technologies such as computer vision, and in this case, semantic segmentation, to apply this to this field. So here again, as we're saying, we're using segmentation. Now in segmentation, what we're doing is we're taking image voxels. And a good way to think of voxels is if you have a 2D image, there's going to be pixels within that image, right? So each pixel has an x-axis and a y-axis. Now for voxels, it's different because now it's 3D pixels in a way. And so it has an x-axis, it has a y-axis, and it has a z-axis. And so what we're doing is we're grouping the image voxels into labeled regions. And for semantic segmentation, every single voxel in the image is classified as a label. So in this human platelet tissue sample that uses electron microscopy, there's 7 different class labels. And it doesn't really mention this in the paper, but in other papers about this data set, there's so many different ones, including the background, the mitochondrion, as it says here. But essentially, we're taking every single voxel and we're classifying it as a certain class label. And then once we put them all those aggregations together for every single voxel, we then get an output that uses semantic segmentation. So here we're introducing a dense semantic segmentation task, which is trying to segment the entirety of the image volume of those human platelet tissue samples with many class labels. To be specific, it's 7. And also, as I mentioned, you wanted to get a lot of insight, right? We wanted to understand the subcellular structure with these tissue environments. And that's really the main reason why this project is being implemented, although it is pretty cool. And that's also another reason. So this project is really impactful, and the biomedical imaging field is rapidly advancing with these sort of projects. So they do say that automating the dense segmentation of EM images is challenging. And the reason for this is because, again, these biological structures are at a nanoscale and they're so complex that it's hard to tell between the features because of the large amount of noise that they have, because of the low amount of contrast. It's hard to differentiate between different cells and different organelles because of the, again, multi-scale features, the noise, and the textural content, which is a really open problem for biomedical imaging. Now, the field of connectomics, which is absolutely interesting, has had more advancements in terms of biomedical segmentation, especially within 3D segmentation, because there's many 3D biomedical segmentation algorithms that are implemented within connectomics as well. But in this paper, they're introducing a neural network that has separate 2D and 3D prediction modules, and they're based off of current modules that are used in volumetric image segmentation, but they're also adding some new things. They're like combining models, so it's like kind of a hybrid multi-model, as you might say. They also talk about some of the challenges they face and how it outperforms baseline and status quo models. So in terms of prior work, the field of 2D semantic segmentation, again, as I mentioned, like there has been so much cloud in that space, like it's advancing so fast. And so a really big part of this prior work was UNet, which has, again, as I mentioned, it has the encoder and the decoder. So the encoder trying to find the where, what of the image, the classification, and the decoder trying to do the precise localization, so the where of the image. And basically, because this UNet, I mean, UNet is 2D and 3D, but over here, like we know that it's talking about 3D segmentation, so it could be referring to each one. Although we do know that the advancements in 2D UNet went and impacted 3D UNet, and then from there, there were so many developments in this space. Now, for this paper, what they did, they used the UNet model, they used the Deep Lab model, and they used the Deep West model. So there's like different parts of it that are really interesting. So what I found in the Deep Lab was that there was spacing. It's called altruist convolution, which is also dilated convolution. And they do this to preserve spatial features in the CNN while keeping the size same. So they're basically changing up the dilation rate, which is the spacing between the kernels, and I actually found that really interesting, honestly. I was just learning about new models because I used UNet for my past one, but this one I didn't really know about. And so along with the altruist convolution, it also does use altruist spatial pyramid and pooling. And so from there, they concatenate them all together and they used a one by one convolution layer to get the output. So I found that really interesting because they're using all of these existing models and basically making a supermodel for this data set. We then look into the methods of how this data set was obtained, because obviously data collection is such a big part of any AI task you do, because if your data is not good, you probably will not get you might get good results, but they probably won't be that factual. Like you just need to make sure that your data is clean, that there's not that much bias, like it's a variety of patients that you're taking it from. Now in this case there were two patients, which honestly did concern me a bit, but they did, again, they used data augmentation and other techniques to increase the size of the data set. So it worked out in the end, because I'll tell you, in the end, the accuracy was pretty good. So essentially, it's saying that this is the data set name, by the way, it's SBFSEM. And SBFSEM is basically, I forgot the acronym. I could try it on the spot, but it probably wouldn't be correct. I know it's like serial block phase serial electron microscopy, something along the lines of that, but it's essentially an electron microscopy technique. And so these image volumes is basically the technique that they use to get the samples from the two humans. And then, as I mentioned before, there were lab members that manually segmented it, and then they also were checked and validated by other professors to make sure that they're valid, because again, we need to make sure that the data is clean and pretty good, because we need the annotated data, right? That was part of the subset that we talked about, so that's also a very important part of it. And it also does say, again, we had 7 classes, as I mentioned, to analyze the structure, and each candidate architecture that they trained it multiple times with different random initializations. And this is basically like randomly initializing the weights and the other parameters in the model. And since this was trained again and again, I mean, it just made it easier to understand what what models did really well, what didn't. And so we also got to the point that this model outperformed the other ones, and that's how they did it. So the data collection part just talked more about like what the data sets consisted of. So this is like the image size of patient one, and it just talked a bit about voxels and like the subvolumes, stuff like that. I didn't want to get too technically deep into it, but oh, here it does mention the 7 classes, so I'll try to read them out with my amazing pronunciation. So background cell, mitochondrion, canacular vessel, the alpha granule, the dense granule, and the dense granule core. So those were the 7 ones that would be semantically segmented within the electron microscopy images. So now we're on to the model, and for this what I'll be doing is I'll actually use this picture right here of the model. Now the thing is, first of all, you can't see my face, so I'm sorry about that, but also for some reason the figure is not actually used in the research paper. So I got a bit confused by that, but it was there in another research paper, and I can link this research paper too. You can see it's right here. So let me explain the architecture behind this model. So it's a 2D to 3D plus 3 by 3 by 3 model. And the composition of it is, first of all, a 2D unit style encoder and decoder. So this is the encoder right here. This is the decoder right here, as I mentioned. And to talk a bit about that, they first of all adapted the 2D unit style encoder and decoder to work on the 3D data. So you see that this has 4 dimensions. Now, if you were working with 2D images, they would have 3 dimensions because they would have a width, they would have a height, and they would have the number of channels, which in this case is right here. Now in 3D images, you have the width, you have the height, and you have the depth plus the channel. So in a way, there's 4 dimensions, but they're technically 3D. So from there, what you're doing is now you have over here, when you have the image, you're starting off with the 3 by 3 by 3 convolutional plus the ReLU activation function. And then from there, you're doing 1 by 3 by 3 convolutional plus ReLU activation function. So this is what happens. You, first of all, increase the amount of channels, but decrease over here when you have the max pooling layer, you decrease the image size by 2. So there's like a proportion of 2 here, as you can see, doubling and then divide by 2. And then you do that a couple of times. And then again, this is the 1 by 2 by 2 max pooling layer. And then once you're done with the encoder, you go through the bottleneck, and then you go to here, which uses a convolutional transposing layer, which is essentially the opposite of max pooling. So it's now increasing the image size. So you can see here it was 37 by 37. Now it became 74 by 74. Now it uses 1 by 3 by 3 convolutional plus ReLU, and then now we have that output, and then it does the same thing again, and we have the convolution transposition layer, and then again we have some basically the opposite to this. Now aside from this, we also have skip connections, which is basically helping with the precise output because we're concatenating information from the encoder to the decoder. Now I'm explaining this part right here for those especially who haven't heard of 2D unit before, because the architecture is pretty much the same except they're adapting it to 3D. Now, aside from the 2D unit model, then we also go from the 2D output and class predictions to form an input to the 3D spatial pyramid module. So this is the 3D spatial pyramid module. And in the 3D spatial pyramid module, you first have a 1 by 2 by 2, here, 1 by 2 by 2. So, oh yeah, we have a 1 by 2 by 2 max pooling layer over here. So this is the max pooling layer, and we have 2 parts of this. So we have the scale 0, which is the input, and we have this, we have 1, which is the pooled. So we have a 2-level spatial pyramid over here. But just note, like if when you're doing this, you have a 1 by 1 convolutional output, and then here we have the residual connection, the skip connection, and then here we have the softmax. So once we have these two, the scales input and pooled, we have pyramid elements that are basically separately passing through the convolutional blocks, and one block is upsampled and added to the block 0 with an output of the residual connection over here. So you can see that right here, right? We are using 3 by 3 by 3 convolutional plus ReLU activation functions 3 times. So this is basically more feature extraction, because you see that there's no max pooling layers, and over here we go from 5 by 5 by 256 296, my bad, to 64 channels, and then 64 channels stays the same. again, residual connection. The main point here was we wanted to have this over here just so we could do more computations and have a more precise output. And then over here, so this part is the pooled, because we're using a 1 by 2 by 2 max pooling layer. And then we're using 3 by 3 by 3 convolutional plus ReLU. And you can see here, the number of channels staying the same. Then we also do have 1 by 1 by 1 convolutional plus ReLU. And then from there, you're doing a transpose layer. So it's going from 256 to 256, 296, sorry, my bad, 296 plus 296 times 5. And then once you're doing the convolutional transpose layer, it goes here into this array. And from there, you're doing a 1 by 1 by 1 convolutional output. And then we have the prediction output. So that is basically explaining the model. And then from there, it started talking about the network training. So we'll go here. So this is more of the math, but it's honestly not that hard to understand, except when you get to some of the formulas, which we will also be getting into. So we have classes from 0 to 6 because, again, we have 7 classes, and I don't know if you're familiar, but code usually starts with 0, so they probably chose it like that. And for every voxel, we have a shape of, with the z-axis, the x-axis, and the y-axis, as I mentioned. And so it keeps talking about how over here, for every single voxel that's part of a certain class, we have, this is the ground truth label for voxel V. So this is the voxel, and then again, class for 0 to 6. And then this is talking a bit more about the shape. So we know that the shape, as it mentions here, like it ends off with 7 channels. So that's what it's referring to. Along with the x, y, and z axis of the voxel, there's also 7 channels. Now, after that, what does it talk about? It also talks about, the per voxel probability. And so it's basically using argmax, which is like an activation function, to find the maximum value of those probabilities and see, well, now, which class does it belong to? And so that's basically how they train it. So from there, what they did was they used a random number generator, and they wanted to use random initializations, which is basically the practice of using random numbers to initialize the parameters in the model, which could be weights, it could be biases. It's basically a bunch of parameters. And so they're using a random number generator, RNG, to control the trainable variable initialization and the training data presentation order. Now, there were also, along with this architecture, there were many other experiments that they did to ensure like what the performance would be without some of the features. So for example, they didn't have 3 by 3 by 3 convolutionals, they didn't have multi-loss 3D, the 3D spatial pyramid that we had, and they also experimented with the original UNet that I mentioned, the 3D UNet thin, the 3D UNet thick, DeepLab plus DRN, and we have DeepLab plus ResNet 101. So they calculated the training for every single one of them, and then they used MIOU, which is basically mean intersection over union, and it's basically a metric score to find the accuracy, and I believe there is also a formula for that as well, and it does talk more about how they calculated it. So here it talks about how it trained 26 instances for 40 epochs, and that was 33,000 steps. And then they just talk about like the GPUs that they used. And from my understanding, this model might have taken a long time to train because of the amount of experiments they did. Now, obviously, I might not have that much computation GPU, just like bringing it more into like subjectively. I don't have that much GPU, but I'll have to look into things like how can I decrease the voxels and the image size of the 3D images so it can fit with my computer, because I'd probably have to use Google Colab or some sort of software that has a high number of GPU. So after this, what it does talk about then is the formulas. So now we're here at the math. Perfect. So first of all, it does say that all the training variables were initialized from the Xavier uniform distributions. And so I probably have that right here. Right. So the Xavier initialization, what it's doing is it's setting layers weights to values chosen from a random uniform distribution between the bounding between this, the plus and the minus. So NI is basically the number of incoming neural network connections. So if you've ever seen an artificial neural network, if you haven't, feel free to search it up now because it'll be easier to understand. Whatever nodes the networks are coming in, whatever the connections are, the network connections attaching to that specific node, like the amount is NI, and then NI plus one is basically the outgoing. So from that layer, with the layer which has all the nodes in the artificial neural network, the outgoing network connections is basically Ni plus one. So from there, the reason why they did this is because then the variance between the weight gradients is basically much smaller for random initialization of the weight. So because if you compare them, this was like without, and then this is with, and this is without. So you can see like the variance is like all messed up, like it's not close at all. And then you see here, they're pretty close together. And so that's why like they wanted to use like Xavier uniform distribution. So after that, they also used a cross entropy loss function. And this is talked about over here, except this is a bit more complicated. So again, they used a regularized weighted sum of cross entropy functions. And so the trainable variables were split up into four subsets. So there's one for the 2D encoder decoder module. There's one for the variables in the 3D spatial pyramid module. There's one for the single 1 by 1 by 1 convolutional variable. And then there's also what's the other one? Right. And the single 1 by 1 by 1 convolutional variable that produces a final 3D class prediction from the spatial pyramid 64 output features right here. 64 output features that are right here before the 1 by 1 by 1 convolutional. So this is basically showing the relationship between whatever I talked about here and this, except all in formulas. This is find the loss function after the training. Now we also have this variable called lambda, and it's basically using regularization techniques such as dropout so that you're getting rid of the unnecessary things in the network, and so it helps it train faster and more efficiently. And so these are the parameters that it uses for regularization. And then over here we have the voxel-wise cross entropy loss function. If you've ever seen a cross entropy loss function, then you'll probably understand what it is. If you don't, like, feel free to search it up. But essentially, it's trying to say that this right here is for all the 7 classes in the data set, in all of the images. And then it starts at j is equal to 1. And this is representing, it is basically representing the voxel that it's at right now. And from there, it's basically finding the cross entropy loss essentially. So it's nothing too advanced. It might look that way, but it's just a bunch of formulas. If you simplify them, like they're not that bad. Now after this, we go into the weighting, the weights of the weights, right? The array of the weights of the nodes and all the neurons in the convolutional layer. So what it's trying to do here is it has a formula, right, for the array of weights. So it has W, right, the initial W that sets a constant for the minimum weight value because it can't be negative. We also have the class balancing term. And the class balancing term, it's basically balancing classes with highly unbalanced distributions because an ML, a machine learning classifier, tends to be more biased towards a majority of class. So over here, like this is a balanced data set. This is an imbalance. So obviously, I mean, it's going to lean towards one data set, and we need to make sure that it's balanced. And because of this, that's why we have this term right here. We also have WEP, which it does talk about as well, and it's an edge preserving term because, as we mentioned, when we have nanoscale images, it's hard to differentiate between different cells and organelles, and so this just makes it a lot easier. We also, again, use data augmentation, which basically modifies the images in the data set and then adds it to the data set so that you have more training data because, again, they had 2 human samples, so obviously there was limited data available. They also did a lot of augmentation with things like the shift in brightness, the contrast, the elastic deformation, and it does talk about that too over here. I came up with this visual. This is the elastic deformation. And aside from that, it also used a decay rate of 0.75. And it should probably, yeah, this is the visual for this one. And basically, you decrease the learning rate exponentially as the amount of epochs increase, as this graph shows. So from there, we had the validation and perform metrics. And this was, again, what I was talking about with the IOU, which is the intersection over union, sorry, intersection over union. And you have the mean IOU. And essentially, it just talks about how they calculated that. This is the formula. I won't go too depth into it, but since it's mean, you know that it'll be divided by 7. And this is finding the IOU, which is adding up all the IOUs and then dividing by the number of classes. That's essentially it. And then they started talking about how the performance of this 2D to 3D plus 3 by 3 by 3 network was compared against those 3 ablations that we talked about. At the top of my head, I don't remember exactly all of them, but there was the multi-class thing. There was no 3 by 3 by 3 features. There was also 5 baseline networks that they found. And it basically overall, like, I don't want to talk too deep into like what every single one did and what exactly they did, but they did say to determine which one is superior to another, they train instances and compare them together. And so the sources of randomness basically really helped to find what the final performance metric scores were for the MIOU and then differentiate those as well. So from there, they found, and you can actually find all of this data in like source code over here, which I found this is why I wanted to do the project because it's open source and many healthcare data is like limited because of the government and then it's mostly private. So this is a really good project if you want to get into dense cellular segmentation or 3D segmentation. So I don't want to go too much into the results, but it overall just said that it really outperformed. Our over here it should say our top 2 to 3D plus 3 by 3 by 3 model performs best on training data, scoring 44.6 MIOU, and then the next one gets 42.1% MIOU. And so it like really increased the segmentation results. And for the 2D unit, it basically improved the MIOU by 20%, which is pretty good. And so it talked about like the ablations and the baseline networks that it used and how it compared. And then it just basically just talked more about that. So in terms of that, like the model really outperformed. I mean, for the first cell, it had an MIOU of 0.54 relative to the other, and the second cell, it was 0.57. And so it indicates two main things. Two main things. First of all, the process for human segmentation in complex biomedical data sets, they need further improvement, right? 0.57, it's like pretty good, like compared to other baseline, but you can do more. So they're just saying like, although this is really impressive and all, there's obviously more potential in this area. That's one thing. Another thing is while algorithmic segmentation errors may differ from the human ones, right? The overall quality approaches basically it literally approaches or exceeds the labelers, and it also again saves time, it saves resources, it saves money in the long run because I mean if we automate this process, I mean if we're getting a good accuracy at the end of the day and we're saving money, that's such a good investment. So that's what it's talking about, those two main things. It might not exceed human quality when it's exposed to new data sets, but when training and when you tested it, I mean it did exceed or approach human labelers. So that's what I talked about. Now, in this work, this is to summarize it all up. We used dense semantic cellular segmentation of 3D electron microscopy images for biomedical imaging, and they used a human platelets data set. So they did say that although there might be challenges for human and algorithmic labelers, automated methods are basically approaching and even exceeding the performance of trained humans, and they might even be integrated into annotation software for the productivity improvement of these humans. Because, I mean, again, trying to make a profit, trying to save money, resources, all of that will help in the end. And so by introducing this hybrid 2D, 3D convolutional neural network architecture, they demonstrated that it performed well and exceeded the expectations between the status quo and the baseline networks. And with the ablation, we know which model and network would work best on the platelet segmentation task. And so they also have a very novel architecture. And so overall, I just really found this valuable. And again, as I mentioned in the last line, they just want to spur the research and development of techniques to make large scale electron microscopy images more useful to system biology as a field, because there's so much potential here. We saw that just from this model, like it did so well compared to baseline networks. Now look at the potential of that. If we try to enhance more and more, if we make more developments, especially with the rapid pace that computer vision and semantic segmentation and 3D segmentation, then cellular segmentation, all of this is at, I mean, from the looks of it, we're in pretty good hands with automation. And although like people might say, oh my God, like robots are going to take over, in this case, like in healthcare, the way we think of it is like it's aiding humans. Now over here, it's approaching and exceeding human labelers. So if humans can be aided with this technology, like it would make such a big difference in terms of annotation software as well. So I hope you enjoyed this research paper, and be sure to like and subscribe.